Thank you. Okay. Thank you so much, Shannon, for the kind introduction. I am thrilled to be presenting our published as well as unpublished work to this audience. So thank you for giving me this opportunity. So before we jump into my talk, I just have one conflict of interest disclosure. We have a patent pending on some of the work that I will be presenting today. So with having said that, so I am a platelet biologist by training, as Shannon mentioned, and I continually to be excited and fascinated by what platelets can do. So these are very small cells present in our body. They're about 2 microns. They do not have a nucleus, yet they pack quite a punch. So platelets have been shown to release a bunch of cytokines, which allows them to interact with immune cells such as monocytes, neutrophils, T cells, and B cells, and platelets in this way can participate in contributing to inflammation. This is beyond their classical function of hemostasis and thrombosis for what we know platelets to be important for, and we know thrombosis and hemostasis both are relevant in contributing to cardiovascular diseases and stroke. In conditions such as diabetes and metabolic dysfunction, platelets are disproportionately affected. And not only are they affected as a downstream consequence of diabetes, but they also participate in contributing to bringing about diabetes as well as other metabolic dysfunction. Platelets also release growth factors, essentially the same way they release cytokines. And this growth factor release has been shown implicated in angiogenesis as well as wound healing. And recently, over the past decade, there has been a lot of interest in how platelets can regulate cancer metastasis and growth of tumor cells. So all in all, platelets are, they're able to pull off this portfolio of diverse functions even without having a nucleus. And that is primarily down to two things that platelets can do remarkably well, and that is endocytosis and exocytosis. As you can see, there is a lot of granules present within platelets. There are these endosomal compartments, your standard early endosomes, late endosomes, multivesicular bodies. There's also, specific to platelets, three classes of granules called the alpha granules. Let me get my pointer on. Sorry. Three classes of granules. So we have the alpha granules, we have dense granules, and we have lysosomes. And these three classes of granules have a lot of bioactive molecules, growth factors, cytokines, chemokines, as well as ADP and ATP, which are important in regulating platelet function. And endocytosis, which is the intake of cargo from the extracellular milieu, and the ability to traffic them into different compartments allows platelets to regulate receptor levels on their plasma membrane as well as on their granules. This allows platelets to also regulate what cargo molecules are trafficked where and how they're loaded individually within these granules at the megakaryocyte level. They also allow platelets to spread on surfaces, such as when there is a breach in the blood vessel and collagen is exposed. One of the first things that platelets do is sit on that exposed collagen by a process called cell adhesion and spreading. And so all of this becomes the first steps in forming a thrombi. Central to this is not only how things get in, but also how things are released by platelets. And so cargo within these granules, as well as endosomes, have to be released. And platelet secretion is a regulated process where only upon agonist stimulation by receptor engagement does these granule contents get released. And this is one of the most well-studied phenomena in platelets, and identifying novel regulators and how we can target those to develop better antithrombotics and antiplatelets is probably been one of the biggest continuing areas of investigation in the platelet biology field. And so one of the questions that I have always been interested in is all of this is happening under steady state condition, but is this process dynamic and can you regulate these processes under settings of various stressors? And so that was one of the questions that I asked when I joined Matt's lab is this process dynamic and can it respond to things like, viral infections or bacterial infections? And so over the past almost 15 years, the Utah Thromboinflammation Group has been trying to understand how platelets and megakaryocytes can respond to systemic inflammation. And what we've been able to show is that, yes, systemic inflammation does reprogram megakaryocytes, not only at the transcriptional level, but also at the translational level. And what it does is it not only modulates the function of megakaryocyte, but it also modulates the functions of the platelets that they produce. So, and we've gone on to use techniques such as genome-wide platelet transcriptomics and ribosome profiling to identify new regulators of not only MK, but also platelet function that are differentially expressed during the various settings of systemic inflammation. One such regulator that we got interested in is this interferon-sensitive gene called IFITM3. It's a big name, so I'll just say it at the get-go. It's called the interferon-induced transmembrane protein. It's a huge family of ISGs. The one that we're specifically interested is 3, and we'll get to that in a minute. But IFITM3 is markedly upregulated in platelets. And this was really dramatic. We found in platelets that were isolated from dengue patients compared to healthy platelets, IFITM3 to be one of the top 5 most upregulated genes in platelets. We also found similar observation in platelets from influenza patients, as well as COVID-19, which we showed recently. And then something that I have contributed to is how IFITM3 is also regulated in a non-viral septic model as like cecal ligation puncture and experimental sepsis models. And so one of the things that was really striking to us about finding IFITM3 to be so high in platelets is that platelets don't express any interferon receptors. Yet, whatever changes are happening at the megakaryocyte level is stable enough that the platelets that they produce do have really high levels of IFITM3, not only at the mRNA, but also at the protein level. So sepsis is something that our lab studies and has been for a long time. It's a global public health burden. More than 30 million people worldwide get sepsis each year, and at least 6 million of these people die from sepsis. So pertinent to our interest, platelets contribute to sepsis and also get affected by sepsis. In fact, loss in platelet counts, which is called thrombocytopenia, is an important predictor for adverse outcome and mortality in septic patients. So we were all being interested in not only what platelets do in sepsis, but specifically what IFITM3 in platelet does in sepsis. So IFITM3, when we look at the mRNA at the transcription level, IFITM3 is one of the top 5 most upregulated genes in platelets from septic patients compared to healthy donors. And this enrichment is also translated at the protein level, where when you look at the protein, healthy platelets don't express IFITM3, but platelets from septic patients express almost 40-fold levels enrichment of IFITM3. And so we were interested in not only what platelets are doing in sepsis, but also what IFITM3 is doing in these in septic conditions. So sorry. So IFITM3 has been classically studied as an antiviral immune effector, and that's largely down to this amphipathic helix that IFITM3 has in its central region, which allows it to prevent viral fusion at the plasma membrane level. And so this is how IFITM3 confers antiviral activity, and this is true not just in the case of COVID-19 or influenza, but across a diverse different viral infections. We weren't necessarily interested in this bit. We were interested in the presence of this YEML motif, which is a classic hallmark for endocytosis. And we wondered whether IFITM3 could regulate endocytosis, given that it's present on endolysosomal membrane compartments in other cells. And so this was the question that we posed, is whether IFITM3 in platelets is regulating endocytosis. And so we first looked at platelets from septic patients, and we compared these with age-matched healthy donors. And when you're looking at these platelets, what I want you to focus on is we fed Alexa 647 labeled fibrinogen to these platelets, and platelets are able to take up this Alexa fibrinogen, which is a marker for clathrin-mediated endocytosis. It's one of the most well-studied. It's one of the most well-studied markers of platelet endocytosis, as well as in MEGS. And what we were able to show is that upon sepsis, or in platelets from septic patients, levels of IFITM3 and fibrinogen are markedly enriched. And not only that, IFITM3 actually co-localizes with fibrinogen, as shown here in these platelets marked by arrowheads. And so we were able to quantify that. And as you can see, fibrinogen levels, endogenous fibrinogen levels, are increased in septic platelets, and as shown here and here. And so what does fibrinogen endocytosis mean in terms of platelet function? When you think about it, the first thing we look at is platelet aggregation. And increased fibrinogen, or fibrinogen by itself, facilitates platelet aggregation because it binds to one of the most important receptor, which is alpha-2b beta-3, that regulates platelet aggregation. And so when you isolate platelets from healthy donors and septic patients, and you treat them with collagen to look at aggregation that is measured by light transmission, you see that the percentage of aggregation in these septic platelets is much higher compared to healthy platelets. And we've shown this not only using collagen, which binds to ITAM receptors and GP6, but also using an ADP analog that binds to P2Y12 receptors that are present on platelets. And so so this is not a specific signaling pathway related phenomena. It's more of a general phenomena to platelets, where increased fibrinogen is associated with increased aggregation in septic patients. And so we wanted to figure out, how does IFITM3 fit into this? And so we turned to a then available IFITM3 deletion knockout model, where IFITM3, which is highly conserved across mice and human, we were able to delete this entire locus that contains not only IFITM3, but also 1, 2, 5, and 6. So this entire locus of IFITM genes was deleted from this mouse model. So we went on to utilize this mouse model to further understand how IFITM3 and other IFITMs were implicated in regulating platelet function. So one of the first things we did was we noticed that clinical as well as experimental sepsis increases interferon levels. And we utilized the sterile inflammation model, where we injected interferon at a really high dose of 25,000 units per mouse for 3 consecutive days, and then isolated the platelets on day 4. And what we were able to show is that upon interferon stimulation, IFITM3 level increases, but in the knockouts, which is where IFITM3 is also deleted in addition to the other IFITM3, we do not see that increase. So what does this mean? So in terms of fibrinogen endocytosis, we looked at the bone marrow megakaryocytes. We started looking at the megs because the interferon injection that we were giving would essentially regulate the meg expression of IFIDM3. Since platelets don't have IFIDM3 receptors or interferon receptors, we first started to look at the megs and then chase the platelets that were produced by these megs. And so upon interferon stimulation, endocytosis of labeled fibrinogen, this is Alexa 647 fibrinogen, was markedly increased. And when you knock out IFITM3, there was essentially no increase or enrichment of fibrinogen endocytosis, suggesting that IFITM3 is somehow important for regulating this endocytosis process of fibrinogen. So we then wanted to see if, and this is quantified here, so we then wanted to see what happens to aggregation. So ablating IFITM3 in murine platelets reduces interferon-induced platelet hyperactivity. And so in kind of similar to our clinical sepsis experiments, we isolated platelets from these mice that were injected with interferon for 3 days. We treated them with collagen, and then we measured light transmission. And as you can see, interferon response increases aggregation in these platelets compared to the knockouts where interferon response did not substantially increase this aggregation. And this has been quantified here, suggesting that IFIDM3 is important and associated with increased not only fibrinogen endocytosis, but also fibrinogen facilitated platelet aggregation. So all of this is really interesting given the fact that fibrinogen endocytosis and platelet aggregation are important drivers and sort of the first steps in regulating thrombus formation. So that was one of the big questions we wanted to ask is it just happening at the aggregation level, or does it have a systemic effect by looking at thrombosis? And so we turned to this model of ferric chloride-induced carotid artery thrombosis model, where we instead of looking at the platelets, we look at thrombus formation in vivo. And so what we do is we take these mice, we inject them with interferon or PBS vehicle control at 25,000 units for 3 days, and then on day 4, we anesthetize the mice. We look at the carotid artery and we add a piece of ferric chloride soaked tissue paper to look at whether a thrombus forms in the carotid artery. And we can use Doppler methods to monitor it in real time. And so the data that you're looking at is the faster the time it takes to occlude, the higher the thrombus or better the thrombus is. So as you can see, upon interferon stimulation, we get a much faster occlusion time, suggesting that interferon stimulation in the presence of IFIDM3 drives thrombus formation. When you knock out IFIDM3, what you find is that not only is thrombus formation unchanged, but the size of the thrombus was also reduced. And the thrombus appears to be much more embolizable. So with all of the data that we had, given that sepsis gives rise to thrombosis, there isn't a sepsis-associated thrombosis risk, we wanted to test what really happens to the actual septic phenomena. And so we turned to this model of cecal ligation and puncture, where we take IFITM3 knockout mice, and then we induce sepsis by doing the cecal ligation puncture, and we isolate platelets, and we also measure various in vivo metrics. In this model, you can see that IFITM3 levels are also increased compared to sham, suggesting that experimental sepsis also induces IFIDM3 enrichment in platelets in a similar fashion it does in clinical sepsis. And this has been quantified here. So we next wanted to see whether some of the sepsis-associated outcomes, such as platelet immune cell interactions, is also affected in the absence of IFIDM3 or not. And what we find here is that compared to sham, wild-type mice that had CLP show a markedly increased levels of platelet neutrophil aggregates. But when you knock out IFITM3, conditionally, you see that the level of platelet neutrophil aggregates is significantly reduced. And this held true for platelet monocyte interactions as well, where you see similar enrichment in platelet monocyte interactions in sepsis, in the presence of IFITM3, and knocking out IFITM3 reduces the burden of platelet monocyte aggregates. And both of these platelet neutrophil and platelet monocyte aggregates are important metrics of systemic inflammation in mice as well as in humans during sepsis. And platelet monocytes typically give rise to a lot of tissue factor, which drives coagulation and some of the disseminated intravascular coagulation phenomena that we see in sepsis. So we also measured pro-inflammatory cytokines. As we know, those are upregulated in sepsis. And we wanted to determine whether IFITM3 has a role in this process. And so what we were able to show is that upon CLP injury, plasma TNF alpha as well as plasma IL-6 levels are increased in the wild-type mice in presence of IFITM3, but knocking out IFITM3 significantly reduces the pro-inflammatory cytokine burden, suggesting that platelet-mediated or platelet-dependent cytokine levels are particularly relevant to determining systemic outcomes in sepsis. So we also looked at levels of tissue injury. I'm just showing here AST, aspartate aminotransferase levels, which is a marker of liver injury. And we find that upon CLP, levels of AST are reduced these are plasma levels of CST. This is reduced in the IFITM3 knockout mice. We next wanted to see whether microvascular thrombi, as measured by platelet aggregates, is affected in these mice during sepsis or not. So we looked at lung sections, and this is stained with collagen. The red is shown, and then we have DAPI, and the white here is platelet aggregates, which is stained by CD41. And you can see there is pockets of these platelet aggregates, which is representative of microthrombus within vascular beds to be more abundant in the wild type samples compared to the knockout samples, suggesting that platelet IFITM3 drives in part microvascular thrombus formation within vascular beds such as lung in septic models. And overall, we're interested in, does any of it have an effect on survival in CLP sepsis models? And as you can imagine, yes, it does. Loss of platelet IFITM3 is associated with improved survival outcomes in CLP sepsis, suggesting that inhibiting interferon-sensitive genes such as IFITM3 could have beneficial effects in not only regulating the pathophysiological outcomes in CLP sepsis, but also survival outcomes in that process. So in addition to that, we wanted to correlate some of our mouse experimental findings with our human studies. So we went back to our septic patients, and what we were able to show is that platelet IFITM3 levels strongly correlates with markers of platelet activation, such as soluble P-selectin, where that's enriched in the plasma of septic patients, and that strongly correlates with IFITM3 expression. Not only does platelet activation and secretion correlates with that, but also markers of thrombosis, such as a thrombin-antithrombin complex, which is elevated in sepsis and is an independent predictor of adverse outcomes and mortality in sepsis, also correlates with IFITM3 expression, suggesting that IFITM3 in platelets and megakaryocytes is an important predictor of not only the phenomena that's associated with sepsis, but also of outcomes in sepsis. So to kind of summarize what I've shown so far is that we've been able to leverage genome-wide platelet transcriptomics to identify novel candidate genes and further validate their function in disease conditions such as sepsis, as well as influenza, COVID-19. And that has enabled us to identify candidate genes such as IFITM3, which is increased during systemic inflammatory stress, both clinical as well as experimental. And we've being able to show that it is a novel regulator of fibrinogen-endocytosis, platelet reactivity, thrombosis, as well as pathophysiological outcomes, together which comprises the thromboinflammatory burden in diseases like sepsis. And so it's again, as a platelet biologist, this is sort of our bread and butter, but we kept wondering if IFITM3 is important in regulating all of these processes in that are common to systemic infection, are there other cell types where IFITM3 could have similar effects? And the first thing we wanted to test was what happens to the cells that produce megakaryocytes and platelets. So we turned our attention to hematopoietic stem cells. And I kind of had a very big, broad picture question is, does IFIDM3 regulate HSC functions during systemic and persistent inflammation? And so as you know, during steady state hematopoiesis, HSCs are able to self-renew, they cycle, they give rise to progenitors, which can give rise to mature blood cells, which is important for maintaining the entire blood system. However, when we have things such as persistent pro-inflammatory cytokines or reactive oxygen species, stressors such as radiation and chemotherapy, that induces a state called stress hematopoiesis, where HSCs reduce or HSC's ability to self-renew is greatly reduced. And this gives rise to less progenitors, and eventually you have less mature blood cells being formed, overall giving rise to pancytopenia, bone marrow failure that can eventually happen, as well as emergence of pre-leukemic clones, which can then give rise to MDS and acute myeloid leukemia. And we wanted to go back to the literature to see is whether IFIDM3 has any role in any of these events that can happen when the hematopoietic system is really stressed. And so one of the things that came out is that IFIDM3 or enrichment of IFIDM3 predicts adverse outcomes in hematologic malignancies such as AML. So in both event-free survival as well as overall survival, IFIDM3 high patients which had just overall IFIDM3 high expression were associated with poor event-free survival as well as overall survival, suggesting that IFIDM3 or high IFIDM3 expression is associated with adverse outcomes in AML. And this is not just specific to AML. When you look to other cancers via AL, you also find similar patterns where high IFIDM3 expression is associated with poor overall survival. This pattern also holds true for mantle cell lymphoma, where IFIDM3 levels are associated with poor survival. So across three different types of leukemias, you see that IFIDM3 expression can be predictive of poor outcomes. And so we really started thinking that can we inhibit IFIDM3 or use our IFIDM3 knockout mouse models to understand whether there is a beneficial effect to in inhibiting IFIDM3. So before jumping into the deep end with AML, I wanted to look at what's happening at steady state. So I just looked at HSC expression and MPP or multipotent progenitor expression of IFIDM3. And in this hierarchical differentiation tree, what you can see is that the more primitive cells have a much more enrichment of IFIDM3 expression. And this is something that we validated based on the single cell RNA-seq data that LT-HSC or the long-term HSCs, which are the most primitive HSC is important for maintaining quiescence, stemness, as well as self-renewal ability, express IFIDM3 highest compared to the rest of the compartments that we looked at. And so we used our IFIDM3 knockout model system to understand what IFIDM3 is doing in HSCs. And so we just validated that IFIDM3 was indeed knocked out in bone marrow cells. This is just general bone marrow cells, not any specific cell type. And then we looked at the basal frequencies of not just the HSCs, but also of multipotent progenitors. And what we were able to show is that no change either in absolute counts or in frequency in steady state baseline HSC and multipotent progenitor levels. So we kept wondering whether if IFIDM3 is not important basally, does it have an effect when you stimulate the system or stress the system with interferons? And so we went back to our tried and tested interferon sterile inflammation model system where we took interferon, we injected wild type and IFIDM3 age-matched mice for 3 days at a really high dose. We waited for 10 days to allow a timeline which allows in vivo interferon levels to drop down to baseline. And then on day 14, we isolated the bone marrow cells from both the femur and the tibia to understand and look for functional assays. And so in this way, we model stress hematopoiesis in our mice to determine the role of IFIDM3. So when you look at just immunophenotyping data and you're comparing both the frequency and absolute numbers of the long-term HSCs as well as the short-term HSCs, we can find that upon in the wild type, in the presence of IFIDM3, the levels are reduced. This suggests that interferon, which we know can stimulate HSCs out of quiescence and cycle, the proportion of cells that cycle in the knockouts is much lower compared to the wild type. So we think that loss of IFIDM3 protects the HSCs from interferon-induced attrition. And we were aware that interferon will upregulate SCA1 expression on SCA1 positive HSCs and can also induce SCA1 expression on SCA1 negative progenitors. So we incorporated SLAM markers such as CD150 to exclude the contaminating effects of interferon-induced SCA1 upregulation. And so when I talk about LTHSCs, I mean LSK CD150 positive, CD48 negative, FLT3 negative. And given that interferon causes HSCs to cycle, we wanted to see whether loss of IFIDM3 had an effect in the ability of LTHSCs and STHSCs to cycle. So we injected mice with BRGU in the presence of interferon, and then we looked at the different cell cycle phases after interferon stimulation. And what we were able to show is that if you look at the G2M phase, which is the actively dividing phase, loss of IFIDM3 was associated with reduced cycling, suggesting that even in the presence of interferon, knocking out IFIDM3 offered some protection to the LT and STHSCs from getting into cycling. Now we know excess cycling can give rise to exhaustion through apoptosis. And so that was the next thing we wanted to see, whether the attrition that we see in the LT and the STHSC numbers in our wild type was because of increased apoptosis. And as you'd expect, loss of IFIDM3 protects HSCs from apoptosis during interferon stimulation. The proportion of annexin 5 positive cells in the wild type, when we looked at LT as well as STHSCs, was much higher compared to the knockout, suggesting that IFIDM3 protects from interferon-induced apoptosis. So physiologically, we wanted to see there's, what's downstream of not only cycling, but also of apoptosis. And so we turned to bone marrow transplant models where we looked at what happens to engraftment after we inject boy jays with bone marrow cells that have experienced inflammatory stress through in the presence of interferon. And so what we did is we looked at both short-term primary as well as long-term secondary, and we measured chimerism at every 4 weeks from mice that were treated from wild type and knockout mice. Cells were isolated from these mice that were treated with interferon and then mixed, 2 million of these cells were mixed with 2 million boy J cells and transplanted into these mice. As you can see, that over up to 16 weeks in the short-term primary, the I5TM3 knockout bone marrow cells engraft much better in this competitive primary. This pattern is also holds true when you look at long-term secondary, that IFITM3 knockout bone marrow cells just engraft better and are resistant to interferon-induced stress. So this is chimerism. We also looked at other lineages. This is B cells, where we found that loss of IFITM3 is associated with increased lymphoid skewness, where there's more B cell engraftment, as well as more T cells, but the myeloid cell proportion was reduced following interferon-induced inflammatory stress. We then wanted to look at what happens to the LSK cells. And so we isolated sort purified LSK cells from mice that were treated with interferon. And then we combined those 1,000 LSK cells with 1,000 boy J cells and transplanted them into boy J mice and chased chimerism at every 4 weeks following transplant. And we were able to see here a similar trend to our competitive bone marrow transplants, that I5TM3 knockout cells engraft better even at as early as 4 weeks of transplant. Similar to our competitive bone marrow transplants, here too, we see a bias towards lymphoid lineage cells for both B cells. The T cells, there's a trend, but it's not significant. And then the myeloid cells were reduced, showed reduced skewness in the primary LSK transplantation. So all of this together suggests that in vivo, upon transplantation, loss of IFIDM3 is associated with improved engraftment potential in both competitive as well as LSK transplants. So we started looking at mechanistically what this could be, what IFITM3 could be regulating in some of these processes. And one of our first pieces of data was really to do single-cell RNA-seq analyses on IFITM3 knockout c-Kit cells. And what we were able to find is that IFITM3 knockout c-Kit cells have an upregulation of quiescence-enforcing genes and a reduction in cell cycle genes. These are important in driving not only cell cycling, but also maintaining quiescence. And we think that this probably could be one of the ways that IFITM3 is regulating stemness in HSCs. And so current work is ongoing to validate some of these targets and to better delineate the mechanistic landscape of how IFITM3 regulates some of these stemness and cycling processes in hematopoietic stem cells. And so to summarize what I've shown you today is we believe that a loss of IFITM3 in HSCs is associated with reduced HSC attrition and apoptosis during interferon-induced stress. And it's also associated with increased bone marrow engraftment, and loss of IFITM3 is further associated with increased quiescence, reduced cell cycling. All of this together helps preserve HSC stemness. And as we know that being able to preserve HSC stemness, especially those of the LT-HSCs, is really important in facilitating better engraftment and durability of transplants in not only bone marrow failure patients, but also in a lot of other patients getting bone marrow failure, getting bone marrow transplants. So we feel that this could be one ways where we can leverage inhibition of interferon-sensitive genes like IFIDM3 to preserve quiescence and stemness, and help patients with transplant issues. So with that, I would like to thank our lab, both present and past members, as well as Adrian Durance's group, especially Adrian and Rohan, as well as Ramiro for the remarkable help that they have given me and Matt in getting the HSC project started. So with that, I'm happy to take any questions. Thank you.
Excellent talk. Any questions? I have a question.
Go for it.
So, first of all, do you know, so this IFITM3 is like super upregulated in MPNs.
Mm-hmm.
Do you know if, I don't know this and I maybe should, if anyone has ever tried to knock it out like in MPN models to see what happens, A, to like the disease, but also to the platelet behavior, like clots and thromboses and things?
Yeah, that's a very good question, Shannon. So we were able to show that in a lot of MPN patients, there is up IFITM in platelets from MPN patients, IFITM3 is increased. But we never quite followed that up with looking
Okay.
at, functional assays. And I believe there is somebody in New Jersey at Rutgers who is
Oh, is it Anandi?
yes,
Yeah, yeah,
yes.
okay.
Yeah, she's studying platelet function in the context of MPN, and she's been a long-term collaborator. In fact, we actually got the platelets from NPM patients for our JCI paper from her.
Okay,
She's part of the author list, and yeah, she's been looking at the function. We don't necessarily know what IFITM3 does in platelets, but we think it's part of the cohort of regulating endocytosis, aggregation, and because all of that really sets the stage for increased clotting and thrombosis.
great.
And MPN patients also exhibit some of those phenotypes.
Right,
So
right, right. Okay, cool. Yeah, because I think a lot about how, like MPN patient, like PV and ET patients are given interferon as a therapy, and like
mhm.
nobody really knows how it works. But like in theory, this is a target of interferon, right?
Yes.
But so like I just thinking about how, I don't know, I think it would be interesting to see how it responds to interferon treatment, like what platelets are doing in general.
I think it would be really cool to like if there is a model to of MPN,
Yeah,
also what's happening at the HSCs.
exactly. Yeah, yeah. Yeah. We
Because
can we should talk more about it.
yeah, absolutely. Yeah, we have several different models of IFITM3 knockouts.
Awesome.
And so we can do, HSCs, platelets, megs, all of that.
Cool. Yeah.
We'll talk more. Yeah.
Great talk. Great talk. It was super interesting.
I also have a question for you, Meenakshi.
Hey,
Maybe
Anna.
maybe I missed it. It was a great talk. Did
Thank
you
you.
also characterize the IFIDM3 knockout at steady state hematopoiesis? Did you show those data or did I miss them?
No,
Okay.
so when we looked at immunophenotyping, we did not see any change in the absolute counts of LT, ST, MPPs, any of that. But interestingly, when we looked at transplants, and because irradiation will induce some interferon, even if you're not like treating the mice with interferon, we were actually able to show that even under non-treated conditions at the bone marrow transplant, both that second primary and secondary, knocking out IFITM3 also enhances their engraftment potential.
Their engraftment potential. And then
Mhm.
I was wondering if you had considered also performing the transplants with purified HSCs because even in your LSK transplants, because you have more HSCs, right?
Yeah,
Within your LSK. So you're
yeah.
even in those transplants, you are potentially transplanting in more HSCs in those transplants, just as like a one-to-one HSC function.
Absolutely. We tried that. We've had very mixed results.
I see.
And it's just because we were not able to get enough numbers. So, but it's still something that Adrian and I have actually talked about this like considerably, that we should revisit those experiments and get those and see what happens, because that would be great. And really goes down to like how specifically we can drill down IFITM3 function in this.
Gotcha. Gotcha. Okay, great. Thank you so much.
Thank you.
Other questions for Meenakshi? Silence. Okay. Thank you so much, Meenakshi. That was really a wonderful talk. And yes, let's definitely talk more about MPN models, but thank you so much for joining us today.
Thank you so much for giving me this opportunity, though.
All
Thank
right.
you, Meenakshi. That was
Thank
awesome.
you.
All right. Bye, everyone.
Okay. Bye, guys.